Thank you. Chairman Graham, Ranking Member Whitehouse, and members of the subcommittee, thank you for inviting me to participate in this hearing to review the state of the science on the therapeutic potential of marijuana and its constituent compounds, and to discuss ways of facilitating more research in this important area. In light of the rapidly evolving landscape regarding the medical use of marijuana and marijuana-derived compounds, it is important to review what we do and do not know about their therapeutic potential. The marijuana plant contains more than 400 compounds, including over 100 cannabinoids. The majority of the research on the therapeutic potential of marijuana has focused on two cannabinoids, delta-9 THC and cannabidiol, or CBD. Most cannabinoids exert their effects by interacting with cannabinoid receptors, which come in two varieties, CB1 and CB2. These are found in the brain and the immune system, respectively. The euphoric effects of THC are caused by its activation of CB1 receptors. CBD, however, has very low affinity for these receptors and consequently does not produce euphoria or intoxication. Its mechanisms of action are not well understood. There is significant research suggesting the potential therapeutic value of cannabinoids in numerous health conditions. To date, the FDA has approved 3 cannabinoid medications, dronabinol, a synthetic form of THC, and very recently a liquid version of dronabinol, and nabilone, a synthetic that is similar to THC. These are approved to treat severe nausea and vomiting in cancer patients undergoing chemotherapy and wasting in patients with AIDS. However, for the most part, patients across the country are using marijuana plant products or extracts that have not undergone rigorous clinical trials, are not regulated for consistency or quality, and are being used for medical conditions with an insufficient or no evidence base supporting these products' effectiveness. What does research tell us about the therapeutic potential of marijuana and cannabinoids? In March of 2016, NIH convened a research summit to review the state of the science on both the harms and therapeutic potential of marijuana and other cannabinoids. While there has been remarkable progress in our understanding of how the endocannabinoid system works, which holds great promise for new medications development, there remains a dearth of evidence from clinical studies regarding the benefits of marijuana or its components. One of the notable exceptions is the mounting evidence for cannabidiol in the treatment of severe forms of pediatric epilepsy. Within the published scientific literature, there have been more than 70 clinical trials exploring the therapeutic effects of marijuana or its constituent cannabinoids for various health conditions. These studies have suggested potential therapeutic value for the treatment of Tourette's syndrome, sleep disorders, substance use disorders, social phobia, neuropathic pain, and psychosis in patients with schizophrenia. While this sounds like a lot, most of these trials would not meet the rigor required for FDA approval because of small numbers of participants, short durations, or other factors that were not well controlled. However, multiple recent reviews, including one by the American Academy of Neurology, concluded that there is strong evidence for the efficacy of marijuana-derived cannabinoids for spasticity and pain associated with multiple sclerosis, and moderate evidence for THC and nabiximols, which is a combination extract of THC and CBD, also known as Sativex, for neuropathic pain. Additionally, preclinical research studies in animals or cells has provided evidence that some cannabinoids may have antioxidant, neuroprotective, anti-inflammatory, anti-tumor, and anti-anxiety properties. NIH supports a broad portfolio of research on cannabinoid compounds and other possible tools to manipulate the function of the endocannabinoid system. In fiscal year 2015, NIH supported 281 projects totaling over $111 million on cannabinoid research, including 49 projects examining the therapeutic properties of cannabinoids. There is a pressing need for more cannabinoid research related to various compounds, dose levels, therapeutic outcomes, and adverse effects. However, the progress of therapeutics development and clinical trials has been slow. This is due in part to the increased time, costs, and administrative efforts associated with research on these and other Schedule I compounds, as you've already heard, and also the currently limited diversity of marijuana supply for research purposes, and the conflicting federal and state laws which hinder the ability to do research with marijuana from state dispensaries or other sources. Though the research is not yet sufficient to support additional drug approval for marijuana or other cannabinoid compounds, addressing these challenges will facilitate more rigorous clinical research and accelerate progress. NIDA will continue to work closely with other federal agencies to do so, and NIH will continue to support research on the therapeutic and harmful effects of marijuana and cannabinoids. Thank you again for inviting me here, and I look forward to any questions you may have.